Hello everyone and welcome to week 9. What we're going to be focusing on this week is going to be microbiology, but I'm also going to be focusing on how to obtain a throat culture and run a rapid strep test, as those are the tests that we will be performing in the lab this week. So microbiology is going to be chapter 8. And when we talk about microbiology, it's really important that you understand really what that is. So microbiology is going to be the study of organisms which are tiny, unusual, microscopic entities that can carry on a living process. For example, could be things like bacteria, viruses, parasites, fungus. All of those are great examples of what are microbiology and what are the organisms that we'll be looking at. When we're discussing microorganisms, it's important to understand that they are all around us. It doesn't have to just be in something that is infected. They are in our food, so what we eat, our water, what we drink, the soil, in the air. They are on our body and they are within our bodies. Now, when I say that we have some in and on our bodies, it's important to know that those are really normal for us and they're supposed to be there. That bacteria is called normal flora. So for example, lactobacillus is a normal flora that helps our body, and it's a good bacteria for our body. So now that we understand what microbiology is and what microorganisms are, it is important that we discuss and understand how we classify those organisms. So I'm going to look at the bacteria, viruses, fungus, the protozoa, all the things that we just talked about and I gave you examples on. I want to kind of go a little bit deeper into. So let's start talking about viruses. So viruses are really the smallest entity and the smallest microbiology form. They will consist of RNA or DNA. So what we make up, they're made up as well. Viruses need a host cell to live on. They can't live on their own. So they'll live off of what the host cell's metabolic system is and does, and that will help it reproduce itself. Now, because viruses live on that host cell, they cannot be grown in a culture media like a agar plate or a petri dish. We cannot grow them in there because the nutrients found within that agar plate or the culture media will not help the virus grow. It doesn't have the metabolism system that a host cell would. Next, we're going to talk about bacteria. And bacteria are considered prokaryotic, which means that they are unicellular or one-celled, but they do not have a true nucleus or a true nuclear membrane. Bacteria, however, does consist of a cell wall and a cell membrane. And bacteria will reproduce by a process that we call binary fission. Now what that is the bacteria itself can actually be asexual. So what will happen is you'll have one big cell that will split into 2 cells, that will split into 4 cells, that will split into 8 cells, which split into 16 cells. It keeps breaking down and it keeps splitting itself. So if you can imagine, bacteria can grow very fast because of this process. Bacteria does and will grow on a culture media because the nutrients that are found in that culture media help to enhance the growth of bacteria. So after we grow bacteria, you can actually view things such as colonies, which are just mass forms of bacteria clumped together. It's kind of fun to watch that grow and to be able to identify it. Next, we're going to have fungus or fungi. Now, fungus are considered eukaryotic, which means they do possess that true nucleus and true nuclear membrane. Unfortunately, though, fungi lack chlorophyll, which is the items found in plants that help them grow and help them reproduce, gives them that green color. Fungi doesn't have that. So when we talk about fungus, this is generally, they want to grow and they want to reproduce themselves in dark, damp areas. So if you've ever stuck bread up in the cupboard and then a few months later you go look at it and it's got fungus on it and mold growing on it, it's because it's been in a dark area, which allows it to grow quickly. Fungus can grow on the culture media, but only if it has certain nutrients in it. And like I kind of made the comment earlier, the most common forms of fungus that you'll see are going to be mold or yeast. And the last one is going to be a parasite. So a parasite is an organism that lives on a host cell, and they get their nutrients from that host cell. The parasites can be multi-celled or single-celled. So for example, a single cell would be something like an amoeba. I know a lot of you have smaller children and probably have seen the show SpongeBob. The evil character, Mr. Plankton, is actually an amoeba. So when you think about a single-celled organism, think about plankton. A multi-celled a parasite would be something like ringworm, roundworm, or tapeworm. They're very large. They have multiple cells to them. Alright, so those are the 4 classifications of organisms. And now we know kind of how to classify them, and we know how to identify them, and we know what we're looking at, and we know what microorganisms are. But we need to be able to identify the different types and patterns of the organisms that we're looking at. So there are 3 main types of shapes that we will use in order to identify organisms. We have coccus, bacillus, and spirillum. So coccus means that they are round in shape. They're generally going to be found in one single pair, which are then called a tetra, a chain, or a cluster. The most common forms of a coccus that you will see are staphylococcus or streptococcus, so staph or strep infections. Next you have bacillus, and these kind of appear in a rod shape. So instead of just being round, they're more like a rod that you would see. They're going to appear in pairs, and sometimes they can combine with the coccus, and then they become coccus bacillus. Okay? So you need to identify both of them if they were a combination pair. Now, where we see the most common forms of UTIs are the most common form of bacillus bacteria or bacillus organisms would be found in our UTIs, gastroenteritis, typhoid fever, which we don't see too much anymore, whooping cough, or gangrene. And the final form and shape that we'll talk about is spirillum. And that is exactly how it sounds. It appears as a spiral shape, so kind of a whoo. And it could also be seen as round, curved round, or in a corkscrew shape. The most common forms that you'll see are going to be syphilis and cholera. Now, cholera is not as oftenly seen anymore as maybe syphilis is, but those are the 2 commons that you'll hear when we talk about spirillum organisms. Alright, so we are now at the point where we've discussed Chapter 8, and I want to talk about this week's lab. So this week we're going to be collecting throat cultures and running rapid strep tests. This is going to be probably one of the more common things that you will be responsible for doing in a medical office. It's one of the ones I know that I see pretty often when I'm in the working field, mostly during this time of year because the weather's starting to get a little bit cooler and you're starting to get more illnesses, so strep is more commonly going to be seen. Strep throat is very contagious, and it's something that we need to be able to inform our patients. Don't have them share drinks or anything like that because it is so contagious. So what you'll have your patient do is have a seat in a chair, And you're going to almost stand above them because it makes it easier to see down into the back of the throat. You'll have the patient tilt their head back, stick their tongue out, and say ah. Now, the reason we have our patients say ah is because when we do that, it actually opens up the area where the tonsils are so they can be swabbed for this examination. During the procedure, you'll want to use a sterile tongue blade or tongue depressor to hold the tongue down while the patient says ah. The reason is because the tongue's natural reaction is to go back and cover the area of the throat that is being examined. Most of the time when anyone starts to gag or they feel like they're choking, your tongue will go back to try to protect it. So, when you perform a throat culture for a rapid strep test, it's really important that you do not touch the teeth or the tongue, because those two areas contain normal flora, so the good bacteria, and what happens is it ends up diluting our sample so you don't get a true and accurate result. So tongue depressor holds the tongue down so it can't go back into that area. You will also want to collect your sample using a figure 8 pattern with a sterile swab. So what that means is you're going to take your swab and you're going to do a figure 8 motion. Now while you're doing that, you also want to rotate the sample collection swab between your fingers so it coats the entire swab, not just one part of it. You do the figure 8 swab to help keep yourself from touching the tongue and the teeth, but also from touching the uvula, which is the piece that hangs down in the back of your throat. So now at this point we have our sample and we're ready to run that rapid strep test. It's important that you know that strep can be divided into groups that appear from A to O. And this is really going to be according to how their antigenic properties appear when you actually grow this culture out. This week in the lab, we'll be working with the Group A strep test, which is looking for a microorganism called S. pygenes. We are looking to help diagnose strep throat, so we're going to need to look for some common signs and symptoms. So some common signs and symptoms you'll see are a sore throat, a bright red pharynx. The tonsils will have what appear to be white patches or they'll be swollen. Patients will feel very tired and have some muscle aches. And generally when patients have strep throat, there is a very bad breath odor that will accompany that. And like I kind of said earlier, it's really important that we explain to our patients how contagious strep throat really is. So we don't want them sharing drinks or food or spreading the disease. So we really want to make sure that they're washing their hands and keeping kind of to themselves for the first 24 hours after being on antibiotics. All right, so that's really the overview that I have for you guys this week. You do have one discussion post this week based off of strep throat symptoms and what you would do if a patient presented with those symptoms. So please make sure that your initial post is completed Tuesday by midnight, and you need to have 2 responses to classmates Saturday by midnight. Also this week you'll have your final quiz for the quarter. So just like we've done in the past, we will do a review here in class, so come prepared for that. Now, it is also week 9, so that means the final draft of your diversity project and your resume project will be due this week, Sunday, by midnight. So please remember that when we talked about your resume project, we said that you needed to not only submit a final draft to me in the Dropbox, but Deb Slaughter is going to need a copy of it as well so she can start getting you guys ready for externship. So when you send her your email with your resume, please tell her when you will be planning or when you plan to go into externship so she can keep a record of that. All right, guys, have a wonderful week, and I will see you Thursday in class. Bye.